study about the optimal condition. Indeed, the optimal condition of pepsin is around 2, and the variation of this pH was already studied to obtain the higher quantity of antibacterial peptides. However, for this study, for the hydrolysis, we used the same pH value. It was 3, and we modified the pH value, but at the end, just to compare, because actually, when we decide to compare with the chemical method, the conventional one, the final hydrolysase is not pH 3. Usually, we have to increase the pH till 8 or any value higher, and after usually is reduced till a pH neutral before using to any real application. So that was the idea, to avoid this step and or to reduce the amount of chemical region we need to achieve the optimal pH value, the
So
ideal one.
just to make it clear, so did you do the hydrolysis at pH 3, 7, and 10, or did you do the hydrolysis at pH 3 and then change the pH later?
We perform all the hydrolysis at pH 3, and then we change the pH to compare the impact of the pH.
Okay, so that's done after the hydrolysis.
Yeah, after the hydrolysis. Yeah.
Okay. Do I have time for one last question? Okay, so this is more about the application. Like pulse electric field is a technique that need like a really high amount of energy, right? So what would you see the application of like this particular technology? Do you think it's something that's easily scale up to a commercial level if you really need to do it, considering the energy cost?
That's a usual question about this topic. I think for me, the answer is yes, but we have to justify well the initial inversion of this kind of technology. So I think to justify this kind of the inversion for this kind of technology, we have to find different application for the process. That's why this approach is good to foster the valorization of different byproducts rich in protein, because we can apply for several things. We can apply the pulse electric field first to improve the extraction of protein from any byproduct, then to improve the performance of the hydrolysis, then improve stop the enzymatic hydrolysis and improve the bioactivity. If we combine those steps under a same process in one only approach, indeed, I think, yeah, we can justify the inversion of this kind of technology.
Okay, thank you.
All right. Well, thank you, judges. Thank you, Zain. Great job responding to micro questions about methodology up to the big picture about how you actually deploy this into a world that's increasingly concerned about things like sustainability and power usage. I did not see any questions in the chat. I'll check one last time. So I think that will wrap up our first speaker. Thank you, Zain.
Thank you so much.
And we're going to move on to our second presenter. Our second presenter is Snigdha Guha. Snigdha is a graduate research assistant pursuing a PhD at the Department of, or with PhD from the Department of Food Science and Technology at the University of Nebraska-Lincoln. Side note, that's where one of my dear colleagues grew up, so go Lincoln. She grew up in New Delhi, India, and completed both a bachelor's and master's degree in microbiology from the University of Delhi. She then pursued a second master's degree in food science and technology and quality assurance at the University of Reading in the UK. She's currently working on the role of dietary bioactive peptides in modulating metabolic diseases. Her research topic is to evaluate bean-derived bioactive peptides' efficacy to reduce vascular inflammation and related pathophysiology. Besides pursuing her research, she's also a teaching assistant for courses in food chemistry and advanced food analysis offered within the food science department at UNL. Today, she's going to present on the efficacy of Great Northern beans-derived gamma-glutamyl peptides in reducing vascular inflammation.
Good morning, everyone. My name is Snigdha, and the title of my research is Efficacy of Great Northern Beans-Derived Gamma-Glutamyl Peptides in Reducing Vascular Inflammation. So let me start by asking, how many of you have had Great Northern beans before? And if not, let me tell you that these beans have some components which are really beneficial for your health, which I'll show you with my research. So to start with, plant-based proteins are gaining a lot of consumer interest in the US in recent years. And because of that, the bioactive peptides from these proteins are also gaining attention. So what are bioactive peptides? These are peptides which, when present in the food protein, inside the food protein, they are inactive. But once they're released from these proteins by gastrointestinal digestion or fermentation or food processing techniques, they are able to show various biological activities such as anti-inflammatory, anti-hypertensive, anti-diabetic. And even though less potent in their action, they can be potentially used as a preventative alternative for pharmacological interventions by use as functional food ingredients or nutraceuticals. And also they provide a great opportunity to diversify the use of agricultural crops beyond their nutritional purposes. So as we know, metabolic diseases such as cardiovascular disease is the #1 cause of death worldwide. And the pharmacological interventions which are associated with them, they have various side effects and they're very expensive procedures. Therefore, we need to keep looking for a more preventative alternate approach so that we can reduce the number of people having to reach that extreme stage where they have to undergo expensive vascular surgeries, for example. Therefore, my research focuses on bioactive peptides, particularly from pulses, which are particularly from dry edible beans, which are a type of pulses, because consumption of pulses have been shown to reduce the rate of metabolic diseases. Also, these beans contain a large amount of free amino acids and gamma-glutamyl peptides. Gamma-glutamyl peptides are a type of bioactive peptides which form their peptide bonds through the gamma carbon of the glutamic acid instead of the usual alpha carbon, which also makes them resistant to gastrointestinal digestion. And they have previously shown anti-inflammatory effects in gastrointestinal inflammation. Therefore, in my project, I'm working with Great Northern beans, which are the largest grown beans in the state of Nebraska. I have developed and optimized an extraction method where I get a whole extract and I fraction it into two parts, fraction A and B. Both these fractions were tested for their bioactivity, and fraction B was found to be bioactive because of, first, the anti-inflammatory assay, which we performed on vascular endothelial cells, which are basically blood vessel cells. So various doses of the peptides were first pre-treated on these endothelial cells, which was then followed by induction of inflammation using TNF-alpha for 6 hours. And at the end of the treatment, we measured the inflammatory biomarkers. So this is one graph representing some of the results we got. And we saw there was a significant reduction in the inflammation. Second, we looked at the antioxidant activity in a cellular-based experiment as well. And we saw that the pre-treatment of the beans reduced the production of reactive oxygen species as compared to the positive control. Then the third thing we wanted to check was whether these peptides can actually survive the gastrointestinal digestion, which would be important because if we consume them as a dietary intervention, they need to retain their activity. So we performed the gastric digestion on fraction B. And after that, we checked the activities again, and both anti-inflammatory and antioxidant activities were retained. A little bit about fraction B. We did not see any high molecular weight proteins, so it was basically consisting of peptides. And when we did a mass spec analysis, we found gamma glutamyl valine as the most abundant gamma glutamyl peptide in this fraction. So then we wanted to check in particular if this peptide was effective in reducing inflammation as well or not. So for that, we used gamma EV in the same inflammatory assay with 3 doses. We pre-treated the cells and then followed by induction again with TNF alpha. And we saw that the highest dose which we used, which was 1 millimolar, was significant or was successful in significantly reducing inflammation for inflammatory biomarkers such as VCAM, then chemokine, MCP1, among various others. We also tested the in vivo efficacy of this peptide in a mouse model of atherosclerosis, which are ApoE knockout mice, where we supplemented the peptide in drinking water for a total of 17 weeks at 2 doses, 50 and 150 milligram per kg body weight. And we saw that at the end of the treatment period, there was significant reduction in lipid accumulation in the aortas of these mice as compared to the positive control, as well as there was reduction in the expression of inflammatory biomarkers, for example, VCAM1. Therefore, the relevance or the long-term goal of this research would be to develop a bean-based functional food, which would be effective in reducing vascular inflammation and thus can provide a strategy for the prevention of cardiovascular diseases. Thank you for your attention.
Snigdha, thank you for the presentation, continuing a trend of very interesting presentations, and who knew, right? Just the simple little Great Northern bean. Grow quite a bit of those in Idaho, I believe, too. Anyhow, judges, it is time for your questions. Please feel free to start asking, and I'll keep an eye on the live chat.
Yes. Yeah, so very interesting talks. And yeah, the northern beans seem to be quite interesting.
Mm-hmm.
Yeah, so just coming back a little bit to your original fractionation. So you have the beans and then you extract protein, is that right?
Correct. Mm-hmm.
And then you fractionate that into 2.
Yes.
So how do you know that if you have gamma glutamate in those, let's say, fraction A and B?
So after we fractionated, we did a mass spec analysis of the peptides and then did a database search for all the peptides which were present in that fraction. And we found gamma glutamyl valine along with other gamma glutamyl peptides like gamma glutamyl cysteine and gamma glutamyl glutamine. But this was the most abundant one in the fraction B.
So did you assay other gamma glutamyl peptides?
Sorry, what?
Did you do some testing on other gamma glutamyl peptides?
Mm-hmm. Yeah, we did gamma glutamyl cysteine as well. And we saw activity in that one, but we mainly progressed with gamma glutamyl valine since that was our most abundant one. So particularly like for the animal study, we just use gamma glutamyl valine.
Yeah, I mean, you can select it because it's the most abundant, but the most abundant is not always the most active.
That is true.
That's why I'm asking if you have data on the other ones.
Personally, I just did gamma glutamyl cysteine in vitro, but there are I did not do it, but in literature, gamma-glutamyl cysteine, gamma-glutamyl phenylalanine, they're all proven to be bioactive.
Yeah,
But
and then maybe one more question. So you have the gamma-glutamyl valine. Did you try testing just the glutamyl valine itself? With no
we did.
glutamyl?
So we compared gamma-glutamyl valine with normal glutamine, and it did not show any inflammatory or anti-inflammatory effect.
On all your assay?
Just the in vitro anti-inflammatory assay.
Okay. Yeah, and then on the cellular model, so you measure the antioxidant. So what test did you perform?
So we first pre-treated the cells with the peptides, and then we added a dye, DCFDA, which is basically an intracellular dye. So it is not fluorescently active, but once you induce inflammation with TNF and reactive oxygen starts producing, so this dye breaks into a fluorescent compound, which you can measure kinetically over a period of 2 hours. So in a positive control where our peptides were not there, the production of this fluorescent dye, or basically production of ROS, was more as compared to the treatment groups where the production of ROS was lesser in that same amount of time.
Yeah, just maybe one last question to follow on that. So what chemistry is going on to give you the fluorescence in the cells?
I'm not very sure, but I think they are quenching the reactive oxygen species, or they are reacting in a way that they are just not allowing them to produce ROS in the cells. We haven't actually looked at the molecular mechanism for the antioxidant, but that is my guess. Because there are similar studies which also quenches these ROS compounds within the cell. So I'm guessing that would be the, but I haven't.
Yeah, it's true that your ROS is reacting with your DCF and then give you a fluorescent compound that you measure. So the more ROS you get, then the more fluorescent, because it's reacting with the DCFDA to give you the fluorescent derivative because of the acetylations.
Yeah.
Okay, thank you.
Welcome. Thank you.
Thank you. Can I ask a question, Snigdha? So it's a great presentation. I really enjoyed it, especially the slides. So looks like all the students are doing really well in slides, right? Okay, so during the experimental process, you did a simulated gastric digestion, right? And conclude that it's not going to impact to the gamma-glutamyl peptides.
Yes.
But the digestion you did, simulated one without any, let's say, physical chewing and like simulating those functions. Am I correct?
Yes.
So do you expect in a real digestion where there is a physical like chewing, mixing, and all those things, can that be impacted to the stability of those peptides?
Potentially it could, but so from our mass spec data, we also found that these peptides are mainly dipeptides and tripeptides. So majorly dipeptides and maybe like a few tripeptides. So if it's already dipeptide, I'm not sure like if it would be broken down, because most of the dipeptides are absorbed intact as well. So like stability wise, I think it should not affect too much, but maybe there is a little effect overall.
Okay, so if you want to like eliminate that question, what kind of a dilution model you can use in order to get rid of that doubt?
Like dilution model for the peptide?
No, digestion model. Like there is a digestion model that you can actually use. So you familiar with that one?
Maybe like batch digestion and take out.
Have you heard about the TIM digestion model where there is actually physical like mixing and it represents all the physical movements as well?
I have not.
Okay, no worries. I was just going through that direction. So that's okay.
But that's a good point.
Because I'm not saying that it will be like having impact on the digestibility of those dipeptide, but if you want to like answer that question, that would be a good way to like eliminate that doubt, right?
Thank you for that.
Thank you very much.
All right, very nice presentation. I really enjoyed it. The slides were very clear. So I have a question more from an application side, right? The beans are great in terms of health, and then they are not consumed as much as in the other part of the world, especially in northern part, right? So in your process, you are doing extraction in the first step, and I don't know what kind of parameters are there. Is there high heat, low heat, pH? I don't know. So you have these two fractions. So when generally these beans are consumed, they are cooked, right? So my question is again towards the stability, like when they are consumed to get the benefit of these important peptides, how does this processing is going to be impact the activity in the longer term? So if you can just elaborate on that.
Yeah, sure. So in my research, I'm basically grinding the beans and extracting the peptides as it is. So there is no cooking or soaking or anything involved, but that's just my research. But there are other research which has been done, for example, in soybean. So, and as well as other beans. So they ferment the beans and they, for example, they make a meso paste out of it, because fermentation has also shown to increase these gamma glutamyl peptides. And some other studies have also like cooked beans and extracted these peptides. So although my study does not involve the cooking aspect in it, but there are other studies which has actually made fermented products. So for example, when I said we can make a bean-based functional food, maybe I was suggesting like we can, because that is one part of the research we have in our lab, we ferment the beans as well, like how we would do. And because gamma glutamyl peptide, another interesting thing is they give rise to the kukumi flavor, if you have heard. So that's like more of a savory flavor. So those, because we are making a functional food, so it wouldn't necessarily have to go through the same processing way which we normally consume beans. But and stability wise, after these fermentation processes, these peptides are still stable and they have shown activity as well. That is why we want to like push our research towards that area, so that, because it is true, people do not generally eat beans. It's mainly like in Asia and other Middle Eastern, but for example, in America, they don't really consume meat. So maybe producing these functional foods might help them eat more beans and also get those benefits.
Wonderful. One more question along the same line. So
we are at time, Bishnu. So if we could do it super quick or take it offline, maybe. I just want to keep us moving. But excellent repartee. Really enjoyed the presentation, Snigdha.
Thank you.
And,
thank you, judges.
Okay, it is time to move on to our third and final pitch. This comes to us from Jasmine Yang, who is a second-year PhD student in the Food Science Graduate Group at the University of California, Davis. She currently works in Dr. Juliana de Morabel's lab, where her thesis research is focused on developing environmentally friendly extraction methods for black bean proteins and prebiotic-like oligosaccharides. Today, she's going to present sequential fractionation as a tool for understanding the physicochemical and thermal properties of aqueous and enzyme-assisted aqueous extracted black bean proteins. Jasmine, take it away.
Thank you so much for the introduction. As you mentioned, my name is Jasmine Yang, and I'm a second-year PhD student at UC Davis in the Bell Lab. And today I will be providing a brief overview of my research on developing environmentally friendly extraction technologies to produce plant protein ingredients. So in the past few years, we've seen major growth in the use of pulses like chickpeas, lentils, and beans in various food products. Therefore, we're interested in better understanding two extraction methods of commercial interest: the aqueous extraction process, known as AEP, and the enzyme-assisted aqueous extraction process, or EAEP. For both of these processes, we milled raw black beans into flour, dispersed the flour in water at a 1:10 solids to liquid ratio, and extracted at pH 9 for 1 hour. The slurry was then centrifuged to obtain a protein-rich skim fraction in the supernamed. AEP is considered the traditional protein extraction method, while EAEP is a useful strategy that uses the same extraction conditions and setup as AEP, just with the addition of enzyme. And in our process specifically, we used 0.5% of a commercial alkaline endoprotease, which has been demonstrated to improve protein extractability and functionality in previous work in our lab. In our lab scale processes, the AEP and EAEP achieved 75 and 81% total protein extraction, respectively. Beans contain anywhere from 18 to 25% protein, depending on the variety, but there's many types of proteins that are present. And a simple method of protein classification is based on solubility in different extraction media. By determining the physical, chemical, and functional properties of the albumins, globulins, prolamins, and glutamins in beans, we can have a better understanding of the AEP and EAEP protein extracts that contain a mixture of these protein classes. So this being the major goal of our work, we fractionated the proteins with the Osborne fractionation method, which is essentially a sequential extraction of proteins using water, salt solution, ethanol, and alkaline media. And our results from fractionation showed that a majority of the bean proteins are water-soluble albumins, followed by globulins and glutamins, and prolamins made up less than 1% of the total protein in beans. We then assessed key physical, chemical, and thermal properties of each Osborne fraction, AEP and EAEP extract. And first, we see here that both extraction media and proteolysis had a significant effect on surface hydrophobicity. In terms of proteolysis, which I've boxed here, the EAEP decreased surface hydrophobicity, potentially due to refolding of protein fragments following hydrolysis, which can rebury hydrophobic residues that were exposed during hydrolysis. Next, we determined zeta potential at acidic and neutral pHs, which kind of gives us an idea of the surface charge of the proteins. And we found that, again, extraction media had a significant effect on the surface charge, but proteolysis only had a significant effect at pH 7. For both of these properties, we see that the AEP proteins closely resemble the albumin fraction, which is expected for aqueous extracted proteins. And lastly, this is a visual representation of the thermal denaturation behavior that was measured using differential scanning calorimetry. And our results here show that native proteins in bean flour had the highest denaturation temperature, followed by globulins and albumin fractions. However, the AEP and EAEP protein extracts had significantly lower denaturation temperatures, potentially due to partial denaturation from alkaline conditions or mild heating during extraction. Importantly, however, we see here that proteolysis did not have a significant effect on thermal behavior, since the AEP and EAEP had nearly identical denaturation temperatures. Lastly, we explored key functional properties under acidic and neutral conditions. First, we looked at solubility, which is considered the most important functional property. Extraction media did have a significant effect, with prolamins exhibiting no solubility at either pH, and proteolysis also did have a significant effect at pH 4.5, increasing solubility by over 40%. And this suggests for applications in products like protein powders for protein shakes or other acidic beverages where solubility is really important, that EAEP proteins might be more suitable. Next, we explored emulsifying capacity, which is relevant in food systems like a vegan mayonnaise, in which you would need a plant-based emulsifying agent. And our results here show that AEP and EAEP proteins yielded similar emulsifying capacity, so either could be used in this application. And lastly, we explored foaming capacity, which is important for products like plant-based whipped cream. And notably, proteolysis decreased foaming properties, likely due to the excessive breakdown of proteins, which we see in a variety of plant proteins as well. Ultimately, it's evident that both AEP and EAEP proteins possess desirable functional properties, and our results here really highlight the importance of tailoring extraction conditions based on the target functionality in the end product. Thank you for your attention, and I look forward to answering any questions from the judges and audience.
Jasmine, thanks for a great presentation. Pulses seem to be the order of the day. With that, it is time to turn it over to the judges for some questions for our third and final pitch. All right, who's coming in first with a question?
Okay, I'll go with a question. Okay, thank you very much, Jasmine. It's a really nice presentation. Again, slides are great. So probably we need to learn from the students now, right? Okay, so my main question is about the zeta potential of the protein extract that you got. So why, like, what is the reason that you can explain for the difference in the two extraction method?
Yeah, so from what I know, with protein hydrolysis or proteolysis, you may be able to shift the isoelectric point of the protein. So in this case, we saw a significant difference at pH 7. So potentially if the isoelectric point of the EAP proteins was shifted to a higher pH, then it might have been at that like tapering stage where it was the same, if that makes sense.
Okay. So do you expect that changes in the zeta potential will impact your functionality?
Yes, for sure. And well, with our functional properties, while you can't directly correlate zeta potential, I think you really see that functional properties are affected by both surface hydrophobicity and zeta potential and extraction media. So there's a lot of factors that come into play when we look at functionality.
Okay, thank you.
Hi, Jasmine. Such a nice presentation. Your delivery was so up to the mark. And then I enjoyed all the presentations. So with your research in your AAP and EAP, you showed that you have a reduced surface hydrophobicity, correct, if I remember? And then you have an improved solubility. Can you explain on that interaction, how these properties, if there is any correlation at all?
Yeah, for sure. I think there is a correlation with surface hydrophobicity. The decrease that we see with protein hydrolysis could be due to the refolding and reburying of some hydrophobic residues that might have been exposed following proteolysis. So due to that decrease in surface hydrophobicity, then we see increased interaction with water and therefore improved solubility.
Right. So what enzyme addition did to in addition to AEP, right? Was that significant difference between these two? I saw in both of the bars that you saw there was a similar pattern, but what value did enzyme add to that process? Because you tested AEP versus EAP, so how much value was added to the functionality or the other characteristics of the work that you did just because of the enzyme? That's my question.
Yes, so enzyme mainly has two functions. One is improved extractability. So in terms of protein extraction yield, we saw an 8 or a 6% increase, which would be very important in industrial settings. And in terms of functionality, like we mentioned before, solubility is very important. And we saw that 40% increase in solubility at acidic pHs. So in applications in acidic food products, EAEP proteins might be more acceptable for that reason. And also we saw a slight increase in emulsifying capacity, which could have also been influenced by the increase in solubility.
Okay. And one more question. At what scale did you do this study, and is there any potential to scale up this process?
Yeah, so we did this at lab scale, 500 milliliters, but we are currently working on scaling up to a 10-liter scale. And we've seen slight improvements in extractability too, which could just be due to the different geometry of the reactor and the different impeller and things like that.
Okay, wonderful, thank you.
Thank you.
Yeah, so I enjoy listening to your presentation. So that was a very good job. So you are comparing the aqueous extraction versus the enzyme extraction. And you are using those two processes to extract proteins. Yeah, so what enzyme did you use again for in the process for the extraction?
We used an alkaline protease. It's a commercial preparation from Danisco.
So you're adding that protease to, let's say, your bean flour?
Yes, so directly into the extraction slurry, it's water, bean flour. We adjusted the pH and then added the enzyme.
Yeah, so you're basically already hydrolyzing the proteins.
Exactly, during
extraction. You are using a protease.
Yes.
So why didn't you extract the protein, for example, before you hydrolyze, like using instead of a protease, use like a carbohydrate?
Thank you for that question. Yeah, so we did do a few tests with carbohydrates pre-treatment, but the problem with that is you need to cater to the optimal pH of the carbohydrates, which is around pH 5. And for plant proteins, that's around the isoelectric point. So that introduces some possible denaturation during that primary stage. So for simplicity, and also we saw higher extractability by just using pH 9 with alkaline protease in the extraction.
Yeah, I'm asking because you are extracting the proteins and then you are fractionating the proteins.
So these are two separate things. We did AEP and EAEP as protein extraction methods, whereas the Osborne fractionation, the sequential fractionation, was just a strategy to help us understand the AEP and EAEP extracts.
So you didn't do the sequential fractionation on, let's say, the aqueous or the enzyme extract?
That's correct. Yeah, we just wanted to see the different protein classes of the bean flour.
Yeah, I understand, but you did that on the enzyme assisted extract. You use that to fractionate. Is that right?
No, the enzyme, the aqueous extraction process and enzyme assisted aqueous extraction process were from the bean flour directly, not from any.
Yeah, okay, so that's fine then. And then for your fractionations, so how efficient is the fractionation? Like, I mean, how much globulin do you have, for example, in the albumin fractions?
So yeah, that is a good question. Because it was a sequential process and we didn't do any type of purification, we do see a little bit of cross-contamination of some globulins in the albumin fraction. Particularly for beans, the primary storage protein is phaseolin, which is highly glycosylated. So it's going to be water soluble. So we saw in our SDS page gel and in our proteomics analysis that there's globulins in our albumin fraction. But the main reason for this sequential fractionation was just to see what the water soluble proteins were, what the salt soluble proteins were, just so we could understand our extraction processes a little more holistically.
Yeah, I mean, the process that you use is commonly used, so that's how it's used. But I was just wondering, like, when we do that, we say, okay, we have the albumin, and then we say these are the properties of the albumin. But generally, we don't actually quantify how much globulin we have there. So, which is not your fault. Other people do that. We may be attributing the property to something else.
We do have one question from the audience that I want to work in before we wrap up. And that is, what happens with the insoluble compounds at the end of the fractionation, and what kind of upcycling can be done with those?
That is an excellent question. So our insoluble fraction for beans is primarily starch and fiber. And there are applications in our lab that we're looking at with potentially using subcritical water extraction to hydrolyze some of these fibers and produce potential prebiotic oligosaccharides. So the work of my thesis will be kind of looking at a circular economy processing strategy for black beans, in which we get the protein fraction from aqueous extraction, and then we utilize the insoluble fraction to produce prebiotic-like oligosaccharides.
Excellent. Thank you. Well, that takes us to the end of our question and answer for our third and final presenter. So thank you, Jasmine. Great presentation. Well, thank you, everyone. Well, thank you, everyone. We're now going to launch our audience poll. The audience vote occurs on the AOCS annual meeting website. Instructions for how to access that will be available in the chat. The audience vote is critical. We appreciate you guys being here, and it does impact the placement of the top finishers. So there is some audience participation in its true form. We encourage all of you to vote for your favorite pitch. Scores are being tallied now. Our judges are conferring amongst themselves, and we'll be back in a few minutes to announce our first place winner and our runner-up. Thanks. All right, well, welcome back. The judges' scores are in, the audience scores are in, so it's time to announce our winner. The first place winner today will receive a $200 cash prize, a recognition certificate, and a student membership in AOCS for 2023. Our second place winner walks away with a $100 cash prize and a recognition certificate. So without further ado, I'm happy to announce that our runner-up today is Jasmine Yang from University of California, Davis. Congrats, Jasmine. And our winner today is Snigdha Guha from University of Nebraska, Lincoln, holding it down from the center of the country for us. So congrats to both of you. And Zain, thank you for your excellent presentation as well. This was really a high quality day. I do want to thank, in addition to our competition participants, I want to thank the judges for their time and volunteer hours here, and the AOCS Foundation who funded the competition. And most critically, our audience today. This is kind of the start of our annual meeting, so hopefully this just keeps the momentum going through to Atlanta. So thank you much, and you all have a good rest of your day.